The overall goal of this procedure is to record spontaneous spatial exploration by synchronously recording physiological signals and videos from freely swimming fish. This is achieved by constructing a large experimental tank surrounded by a sensory isolation chamber. Next, a multi-channeled electrical recording device is constructed to precisely and reliably measure the electric organ discharge. An infrared video camera is used to synchronously track the animal's body posture and trajectories. Results are obtained that show interplay between an animal's body movements and its active electrosensory sampling rate.
The main advantage of this technique over existing methods, like eliciting evoked responses by delivering external stimuli to restrained animals, is that the spontaneous and naturalistic behaviors from a freely behaving animal can be non-invasively observed. This method can help answer key questions in the neuroethology field, such as the role of active sensing during self-guided exploratory behaviors. We first had the idea of this method when we began to investigate the role of active electrosensory sampling in pulse-type weakly electric fish during self-guided exploration and spatial learning.
Visual demonstration of this method is critical due to difficulties with the movement-induced recording artifacts and the complications associated with lighting and imaging through water.
The chamber rests on an anti-vibration surface containing an electrically shielded heating element over thermally graded foam padding.